Hereby, I open this academic ceremony in which Nina Possemis will defend the academic thesis Diagnostics and Phosphodiesterase 4 Inhibition Intervention within the Cognitive Spectrum, from Healthy Aging to Dementia. Can I please invite you first to present a summary of your thesis?
Dear Prorector, members of the Corona, dear colleagues, friends and family. In the upcoming 15 minutes, I would like to present to you the research outlined in my thesis called Diagnostics and Phosphodiesterase 4 Inhibition Intervention, from Healthy Aging to Dementia. Imagine a city of 300,000 inhabitants, like the size of Utrecht, where everyone is slowly losing their memories. That's approximately the number of individuals living with dementia in the Netherlands. It begins with small slip-ups, such as misplacing a key or forgetting an appointment, but steadily worsens in the end, disrupting daily lives. Right now, treatments for these memory issues are like Band-Aids. They help a little, but they don't go to the deeper problem. People need more than that. They need something that can go to the root of the problem. Two things could change the future of this city: detecting memory problems earlier and discovering treatments that can slow and help memory. My research is focused on both. Discovering early methods of diagnosing cognitive impairment and also treatments that can preserve memories longer. Accordingly, this thesis is divided into two multiple parts. I would like to start with the parts in which we wanted to see whether the test can diagnose early and also predict dementia. Imagine you've been feeling a bit forgetful lately. Maybe you keep losing track of your keys or you forgot an appointment. You decide to talk to your doctor about it, and they suggest a visit to the memory clinic. At the memory clinic, the doctors want to understand how you're doing in your everyday life. So they'll check your medical history and talk to someone who is close to you, like a family member or a partner, to get a better sense of the challenges you're currently facing in your daily life, and to get a better sense of how you're doing in your daily life, such as whether you're remembering if you're taking your medicine or if you are handling day-to-day tasks. Then a neuropsychological assessment is performed, a series of activities that might feel like puzzles or activities. However, they mean so much more than that. Additional measures can include a blood draw or a look at your brain. This is usually with a brain scan, and they might perform a lumbar puncture to check the fluid around your brain. While all these measures can be equally important, I'd like to zoom in on the neuropsychological assessments performed at the memory clinic. But first, what is a neuropsychological test? Imagine it's like a checkup for your brain, but instead of using stethoscopes or a blood measure, we use activities and puzzles. These activities are designed to help us understand how well your brain is working in different areas, like memory, attention, problem solving, and also language. We're living in a world where more and more things are becoming digital, like shopping, banking, and even healthcare. Neuropsychological assessments are no exception. Many parts of this test can now be digitized. For example, during a traditional neuropsychological test, your assessor listens as you speak, takes notes, or scores your responses. Now, with digital tools, we can record audio and use these automatic speech scoring system, which means we record the audio during a test, like a memory test or a language test, and then use scoring system that automatically score how you're doing. After all the tests and assessments at the memory clinic are done, it's time for your doctor to communicate to you what they found. For some people, the doctor might say they have subjective cognitive decline, or SCD. This means you've noticed memory or attention problems, but the tests show everything is still within normal range. You're experiencing symptoms, but they aren't affecting your daily life. For others, the diagnosis might be mild cognitive impairment, MCI. This means the tests found some changes in your memory and thinking skills, even though you're still able to handle daily activities just fine. And then some people might get the diagnosis of dementia. This means there are clear changes in both your thinking skills and how you manage daily tasks. Starting with chapter 2, we were interested in the digit span task. This task has 3 parts. In the first part, it's called forward test. The clinician will say a series of numbers out loud, like 4, 1, 4, 7, 2, and your job is actually to repeat those numbers back in the exact order they were given. In the second part of the test, the backward test, the clinician again will say a series of numbers, but instead of repeating the numbers in the same exact order, you are instructed to say them backwards. You start with the last number and end with the first. In the last part, the sorting test, instead of repeating them forward or backward, you need to sort them from smallest to largest. One goal in this chapter was to see, based on already existing diagnoses, does the digit span, so this task, does it diagnose all individuals accurately? Another goal was to see whether the digit span task was able to predict which individuals in the future would be diagnosed with dementia that were not diagnosed with dementia in the beginning. We found some interesting patterns. The backward and sorting subtasks were particularly good at accurately distinguishing between diagnostic groups. The forward task, however, wasn't as useful for this purpose, probably because it's not very challenging. So, since it's easier, it doesn't show as much difference between people with memory problems and without memory problems. Interestingly, the backward and the sorting tasks were also good at predicting a dementia diagnosis. This makes sense because the more difficult the task, the better it can reveal who is having memory issues and who is not, and also thus predicting if someone will receive a diagnosis with dementia or not. In conclusion, due to the increased difficulty of the backward and sorting subtests, they were able to offer accurate diagnosis and predict whether individuals would receive a dementia diagnosis. In chapter 3 and 4, we focused on another memory test. This test is called the 15-word verbal learning test, and when performing this test, you are given a list of 15 words, and you are asked to remember them. After the words are read out, your job is to recall as many words as you can. Okay, we go through the list again, and then once again, and again, and again, and again. We do this 5 times, and each time you are instructed to recall as many words as you remember. And we do this 5 times because we want to see how well you learn and remember the words over multiple attempts. 20 minutes later, we ask you to recall words from that list again, without any warning. This surprise recall helps us understand how much you still remember after some time has passed. This specific task was investigated in 2 ways, namely in a face-to-face assessment and in a telephone assessment. And this means face-to-face, we invited participants to the memory clinic and we saw them face-to-face and recorded the audio during the task. And via telephone, the audio was recorded anyway through the telephone. So we recorded the audio in both assessments and then used the application on a phone or an iPad that analyzed the recordings and automatically scored the words they said. Out of this task, it's important to look, what does the clinician look at? The main scores used in this task are the total immediate recall. This means all the words that the person has mentioned that were correct are tallied up over the 5 times, and this results in a total correct recall. Another score used to interpret the performance on this task is the delayed correct recall, so all correctly recalled words after 20 minutes. Due to the automatic processing of the audio, and not just the clinician writing down scores, it is possible to calculate way more many features, such as pauses between words, or primacy words, which are the first 3 words of the list, or recency words, which are the last 3 words of the list, and the total duration, for example, of how long someone takes to recall the words. There are way many more. In our study, we investigated approximately 100 additional features. Our first goal was to find out if the technology could score the test as accurately as the human. We compared scores given by the application to those given by a trained professional to see if they matched up. As a second goal, we wanted to check if the technology could tell the difference between people who have memory issues and those who don't. Our last goal was to see whether these additional features, I told you about 100 approximately, that can automatically be calculated would add any additional value in diagnosing individuals. We found the following. Scores based on speech and audio were nearly as accurate as a human counterpart, the clinician. Those scores based on speech showed a fair diagnostic ability, and new speech features, so the 100 more, increased also this diagnostic accuracy. So what now? You might just receive the diagnosis, or maybe you're trying to prevent memory problems. Right now, there are no effective medications that can stop or slow down memory problems. Some current drugs are available, but they have small effects and often come with side effects. To understand how we can improve memory, think about how our brain strengthens connections between brain cells when we learn something new, like practicing a skill until it becomes second nature. This process has a specific name, and it's called long-term potentiation, or short LTP. It's crucial for memory. A tiny helper, also called an enzyme, named PDE4, plays a key role in controlling a substance in the brain called cyclic AMP. Together, these interact with each other and help, therefore, memory. If we can inhibit PDE4, we can increase cyclic AMP and boost LTP, and therefore maybe enhance memory. In part 2 of my thesis, I want to talk a bit more about the intervention-based PDE4 inhibition in healthy individuals, but also in individuals with cognitive impairment. In chapter 5 and 6, we were interested whether one dose, which is 3 capsules of CILTEP, could enhance cognition in healthy middle-aged and elderly. CILTEP, also recently rebranded as Neurofuel, is a nootropic supplement. You can think of neurotropic supplements as brain boosters or smart drugs. That's how they're often also called in the media. They are consumed by individuals to help with things like memory, focus, and learning. At least that's what they promise. So why were we interested in this specific neurotropic? Because of its ingredients. Some of these ingredients are said to act as a PDE4 inhibitor. We wanted to see whether CILTEP enhances cognition in both middle-aged and elderly healthy participants. We also wanted to see whether CILTEP changes EEG signals, so-called brain signals, measured by a cap on your head with many electrodes. And lastly, we were interested whether CILTEP would affect vitals of the participants, such as blood pressure and heart rate. Well, we found that CILTEP, at least one dose, so 3 capsules, had no immediate effects on cognition, so that it did not enhance cognition in both populations. So that means in healthy middle-aged participants and in elderly elderly participants. There was also no immediate change in brain signals. And lastly, there was also no immediate change in blood pressure or heart rate. In the last chapter, in chapter 7, we present a setup of the ROMEMA study. Unfortunately, we have no study results to share with you right now. However, the study has finished 2 weeks ago, and soon we'll be able to share those results with you. ROMEMA is a study where we wanted to see whether roflumilast, also a PDE4 inhibitor, can enhance or stabilize memory in individuals diagnosed with MCI or mild dementia. We think that this PDE4 inhibitor, such as roflumilast, could be used to help specifically individuals with Alzheimer's disease because there are special PDE4 connection points in specific structure in the brain that helps us form memories. In Alzheimer's, this structure is one of the first areas to shrink and stop working properly. By targeting these connection points, a PDE4 inhibitor might help protect the brain and slow down memory loss. This is a placebo-controlled study. That means that individuals were either randomly put in a group that you were instructed to take placebo, which means there's no active ingredient in that 50 microgram or 100 microgram roflumilast. It's also chronic administration, so we instructed individuals to take roflumilast or placebo for 6 months. And our main outcome was the memory test I showed you with the words you had to remember. And also we are very interested in daily activities, mood and well-being of the participants, which is why we're also getting a person that knows the individual very well, such as a partner, and ask them how the participant's daily life is going. So, conclusions. The digit span test, specifically the last 2 sub-tests, the backward and the sorting order, can be of diagnostic and prognostic value. Speech-derived scores are close to accurate as manual scoring. Diagnostic ability of speech-derived scores are good. And lastly, 1 dose, 3 capsules of the neurotropic CILTAB does not enhance cognition in cognitively healthy participants. I would like to thank you all for your attention and give the word back to the prorector.
Thank you very much for this clear presentation. We will now start the opposition, and the opposition will be opened by Professor David Linden. He was chair of the assessment committee and he's a professor at Maastricht University.
Dear candidate, first of all, thanks for this very clear presentation and congratulations on your thesis, also to your promotion team. I would like to start our discussion, our scholarly discussion, and ethical discussion, in fact, probably, with your thesis number 9. And I would like to ask one of your paranymphs to come forward and read thesis number 9, Stelling 9.
The insistence on rigid adherence to research protocols by clinician researchers, even at the expense of immediate patient care, is an unethical approach that prioritizes data over human lives.
Thank you. Well, that is a strong statement, dear candidate. Perhaps you could expand on it a little bit more, what you mean, or whether you have a particular scenario in mind that you're criticizing here.
Highly esteemed opponent, thank you for your compliments and your question. It is a very strong statement. I am aware of that, and that's why I find we need to focus on this. At least in the memory clinic, my role as a sort of clinician, but also researcher, I find it hard to, if individuals have questions about what now, the study is done, we do not give the medication anymore, but they ask for more help. And as my job, it's actually to say, as a researcher, our help stops here. This was the study. This is what you have read in the patient information sheet. I won't help you anymore. I tell you to go to your GP or to someone else who is a professional and can help you further. However, also a little bit the clinician and maybe also the human in me and my expertise make it hard to ignore these help or call for help. And I still give them more information that supposedly is not in the protocol. It doesn't say if a person asks for help, such as an informal caregiver who asks for help, how can I deal with the cognition issues of my husband or wife? It doesn't say that in the protocol, but in the end, I advise programs from especially our university that might help them and continue to give them a little bit more comfort even after the study.
So, do I understand correctly that your main point is about the aftercare, or are you saying there should be more flexibility in deviating from trial protocols during a trial?
I don't think there should be more deviation from a protocol during a trial. However, I understand it's normal if that happens. As the human in us that wants to help, and maybe when they have other questions related to the disease itself, that we help them in case of knowledge, or even suggesting other programs that could help them in that moment.
And would you say, I think this is a very interesting and important discussion, would you say that we're sort of perhaps looking at, because you're saying the clinician, but also I think the insistence is also on the part of, potentially, at least we think, on the part of ethics committees and regulatory bodies, not just the clinicians. I mean, are we taking research trial protocols too much as sort of legal texts from which we must not deviate? Do you think there should be or could be more flexibility?
I think there could be more flexibility. It's a bit hard when in that moment you need to decide whether this deviates truly from the protocol and will change the outcome of your study. That's the difficult moment where you actually need to take, in that moment you need to say stop, I need to discuss this with the researcher of the study, and in this moment this feels very inhumane to say, sorry, I can't right now, I'll come back to you, especially if a person is emotional.
And I understand your point, and also, of course, if you have to wait for an amendment to be processed, that is not the timeline that your patient normally needs. So this is a challenge. In relation to but I think the strongest point that you're making is in relation to the post-trial care, which in fact you are covering in Chapter 7. Because you had provisions for post-trial care in your trial. Can you perhaps explain what you implemented there?
Post-trial care is actually quite reduced. So we do offer just communication still with us. However, we don't offer any study medication anymore. Also, any side effects that last longer than a certain amount are not our issue anymore. So we refer them to the GP in case of any struggles related to cognition and physical aspects.
And would you say that perhaps it shouldn't be the responsibility of the GP, but potentially the institution running the trial, for example?
I think that depends on the issue at hand. What I've heard also on other conferences is that participants often, after they've participated, especially the study is very long, 6 months, and then they're suddenly dropped by a researcher. They feel lonely, they feel they've participated, they feel a bit dependent on us. I would like to think about more how we can make this transition a bit easier. So it is, I think, the GP, obviously, because he's trained for this, so anything related to cognition or side effects or anything physical, I cannot give any advice because I'm not a doctor, I'm not allowed to give advice. However, this small transition of someone that stops the study, just simply dropping them, for me, that's not acceptable.
Okay, thank you. That's very clear. I would like to give the word back to the Pro-Rector.
Thank you. The opposition will be continued by Professor Renate de Groot. She's a professor of biopsychology of learning at the Open University, and she was also a member of the assessment committee.
Thank you. Esteemed candidate, first of all, I would like to congratulate you on this nice thesis. You have delivered a solid thesis around a very interesting topic, not only for science, but also for the medical practice. And of course, I would like to also extend my congratulations to your supervisory team, Ines Raamakers, Frans Verheij, and Arjan Blokland. My questions concern chapter 5 and 6. And first, I would like you to elaborate a bit more on the underlying potential mechanisms as to why and how CILTEP could influence acutely cognitive functioning or not, as you didn't show any effects. And before you elaborate a bit more about the mechanisms, and you actually already did so during your presentation, if I understood correctly, CILTEP consists of many ingredients, and you proposed that CILTEP might influence the cyclic AMP homeostasis. Cyclic AMP is an energy-carrying molecule, and it's used for intracellular signal transduction. But why is it then so important for cognitive functioning? How sure are you that it arrives so quickly really in the neurons? Doesn't it increase the cyclic AMP in the body cells first? So how does it affect the neuron? And if it doesn't arrive in the neuron that fast, I would like to propose an alternative mechanism based on CILTEP. And that is that also glycogen is converted into glucose pretty fast based on CILTEP. And glucose is actually the only real fuel for the brain. But if that's the underlying mechanism, and now I'm probably talking Dunglish, haven't you been a thief of your own results? Because you provided all your participants at the beginning of your experiment with a breakfast. So, a lot of glucose. Can you elaborate a bit upon this, please?
Highly esteemed opponent, thank you for your compliments and also for your question. It's quite a question of multiple parts, so I will actually start at the end. I'm not quite familiar with how glucose could truly impact cognition or the brain. Of course, the brain mainly uses glucose as its fuel, as you mentioned. However, there are also certain diets maybe that say glucose is not necessary and we should be more focused on protein and fats. So that would be still maybe a way of me to say glucose is not necessary for this. But we did give a breakfast in the beginning of the test day because we wanted to avoid that people ate anything that contained lutein because it's a flavonoid, so that's easily ingested like through eggs or dairy. And also not giving them maybe could have caused fatigue. So I think in regards to the being a thief of our own results, or there's no way around it that people need to eat something. And also when they use these capsules at home, they eat something. Usually it's also the supplement. On the supplement it says to ingest it with food or sometimes also without. But I think there's no way around it that we would have given them something. And I think the mechanism through cyclic AMP stays the same. So I am unsure what you mean by the glucose, how it's...
Well, I think we agree upon the fact that it's not by means of the fact that glycogen is converted in glucose. I mean, so you focus on cyclic AMP, but then in these chapters you also report that you measured blood levels. How sure are you that it's really infecting the intracellular mechanisms of the neurons? Because as far as I understand, the neurons are responsible for your cognitive functioning.
Yeah, the best way to measure that would be doing post-mortem samples. However, obviously, we do not want to kill our participants to directly measure that. So measuring blood was the best way to actually get close to that. How sure can we be? We cannot be entirely sure. However, if it would have shown that there is enough of the plasma, then we could have said maybe it is another mechanism. So it was, I'm not going to say it was an ill setup, but if it were up to me, I would have done a pilot study beforehand in which we just, without letting all the participants go through a whole neuropsychological assessment, just drew blood to see if levels could be high enough.
But how sure are you that the responsible ingredients of CILTEP really cross the blood-brain barrier?
Well,
is there previous literature available about that? Or?
Yes, there is previous literature of the two individual ingredients that we were interested in, so artichoke extract and forskolin. Though there has been literature on it that it does reach or go through the blood-brain barrier, and also artichoke extract, which includes luteolin, this is the best PDE4 inhibitor, or the highest you can have. Any other supplements has lower bioavailability than that. But the bioavailability was still very low. So actually, if we would have instructed individuals to get the IC50 of a PDE4 inhibitor, such as roflumilast, we would have needed approximately 7,000 capsules instead of 3.
And how fast would it then cross the blood-brain barrier? So with the design of your study, I think you tested them cognitively after 30 or 60 minutes?
60 minutes, indeed. Yeah. So we expected plasma levels to be at highest at 60 minutes.
But that are the plasma levels.
Yes.
That are not the levels in the which have passed the blood-brain barrier?
No, that is very correct. So that is the only way we actually measured whether it is in the body.
Okay, thank you very much.
The opposition will be continued by Professor Daniel van den Hove. He is a professor of neuroepigenetics at our university, and he was also a member of the assessment committee.
Thank you very much. First of all, my compliments for this very nice thesis. It was a pleasure to read, and I also loved your presentation. Also, my compliments to your supervisory team with this very nice thesis. Of course, some questions. I would first like to start on chapter 7. Of course, you said that the study is still ongoing or was just finished, but the data are not in yet. I first want to go to the dosing regimen that you have chosen and how that relates to the previous study done in healthy old individuals. So you remember which dose was taken there or given there?
100.
Yeah. Now you use 50 or 100. So my first question is actually why, and particularly in view of the fact that the previous study was in an acute setting, so looking at an acute effect, and your study now is focusing on, let's say, long-term treatment with the same compound. So that would be my first question. Why this dose regimen?
Highly esteemed opponent, thank you for your compliments and your question. Indeed, so we chose as a dosing regimen placebo 50 microgram and 100, and as you said in the previous study, it was based on an acute study on 100. So we assumed that at least 100 would be effective for acute. We don't know, we have never performed any chronic studies. So on the basis of that, because we test participants also acutely and then also chronically, so we can compare that. We've also never looked at 50 microgram. So this way is actually that we are sure that 100 will work for the acute and that the 50 chronically will act as the same as the 100 through plasma levels, that they will chronically build up.
Yeah, I understand that rationale. Are you also aware of animal studies using similar compounds? Because they're actually in chronic experiments, they even go a factor lower when compared to acute dosing. So I was wondering whether it's something that you considered to even go lower with the chronic dosing, because then the effects may add up, etc. So is this something you considered? Of course, you have to start somewhere. You can't do this all. So I understand the choice made, but is it something that you considered?
We have not considered that. So we're also very interested in this study and the pharmacokinetics results. And it will be the first time we will have a look at 50 microgram and we will see whether...
Okay. And you mentioned already in between acute chronic. Of course, here you actually also still give the compound close to the testing. So are you then actually measuring chronic effects only, or how do you see that?
We don't give it close to the testing, at least individuals take it at home.
Yeah, but on the same day or just the evening before? Or when did you actually take it? Are they free to take it when they...
Yeah, they're free to take it whenever, so you might have variation in that, but we assume that if a participant adheres to the medication rules that it should chronically be also in the blood, so even if they miss a day, then that's fine, as long as they continue and we don't miss weeks or days, which is why we have a medication diary in which they can say, okay, I took it today or I didn't.
Yeah, and of course, if you, at the day of testing, also take a plasma sample, you could still correct for the levels and relate it to this. Okay, thanks. Now, my next question was actually related to the previous discussion. Not about breakfast, but in your answer, you also said, yeah, if we don't give it to them, or they might suffer from fatigue. Now, you also state here in the thesis that you actually don't allow them to drink coffee, which of course makes sense. But at the same time, if you're a regular coffee drinker and all of a sudden you're not allowed to drink coffee, it might also lead to fatigue or any other cognitive effects. So how do you see that? I mean, I also understand that the opposite is also not really helpful or can introduce bias. So how do you see that? Were there people that actually reported that they would have liked to have coffee or whatever? Or?
I'm not sure. During this specific study, I know during Romema we also asked them if they drank coffee or not, because we want to correct for it eventually, because caffeine, in my honest opinion, is the best neurotropic, if you want to say it like that. But indeed, but at least my opinion is that they were, there was no coffee in both conditions, so in the placebo and in the CILTEP. So at least they're correct for themselves. It's a crossover study, so they are their own placebo. Yeah. So at least it's consistent in that, but it could be that participants' performance was on both days lower than they should be. Yeah.
And I agree that caffeine or coffee is a really nice cognition enhancer, but I think it's mainly when you're not a regular drinker and then you drink caffeine, then it has this effect. If you're a regular drinking, then actually you just need it to perform at normal levels. But okay, that's something else. I'm also a regular drinker, so I understand the issue. Now maybe a final short question. You talk about dropout particularly in relation to chapter 7. I wanted to talk about it. Because you said, hey, we expect 10% dropout based on previous studies, but those previous studies were particularly in healthy individuals or SCD and MCI individuals, and now you actually have early AD cases in there as well. Yeah. Don't you expect, or did you actually experience, now I can ask that, did you actually experience high levels of dropout based on the fact that you're dealing with even more vulnerable individuals?
So my expectation beforehand was actually that there will be less dropout. I know it's a long study, it's a medication study, however, the stakes are higher. Individuals with dementia or mild cognitive impairments, they have higher hopes. This is a sort of last chance. So I thought, I also noticed it, actually, we only had 3 dropouts, of which 2 were adverse events that were possibly or not, maybe not related to the medication, and 1 was a loss to follow-up. So the dropout was actually quite low. And this I also expected because the stakes are so much higher. Individuals are putting their resources and time into this study, and it's very valuable.
Okay, thank you. That's very good to hear, and happy with the answers, and give the word back to the program.
Thank you. The opposition will be continued by Dr. Alexandra Koenig. She is working at the University of Côte d'Azur in Nice, and she participates online.
Yes, hello. Sorry that I can't be there in person. I am at the conference, but yeah, estimated candidate. Also from my side, I would like to congratulate you to this excellent work on your thesis, and also a very great, clear presentation. It was really a pleasure working and collaborating with you also on some of those publications and work you presented. So I have maybe a very obvious question, but let me say, I felt you presented well in the first part of the PhD, focuses on diagnostics and the added value of some sort of refinements of neuropsychological tests. So also I would say more innovative ways, like the automated speech measures that were explored. And I think the reasoning behind this is that we need more sensitive cognitive measures. So I wonder now in the second part of your PhD, where you really look more at intervention studies, none of them actually appeared there. Even though we had relatively good results, but not even, as an exploratory outcome measure, for example. So regarding also chapter 7, in the protocol description, it says that, for example, primary outcome is the VLT. Do you think really that a simple only recall score will be sufficient to detect maybe even subtle changes due to the intervention, due to the compound? And maybe there will be changes that will be missed using the classical way of scoring. Can you maybe elaborate a little bit on this in general? What are your impressions also for future of such trials when it comes to cognitive outcome measures? Is there anything more to maybe use? Also, I saw that there was no remote testing, for example. People have to always come back into the clinic. You think maybe things will be missed here? And since there were daily reporting of medication, why could not have been maybe something remote additionally added on here? So I really it's more about elaborating on all these, please.
Thank you for your compliments and also for your question. I will try to find a focus point. As of why it's not remote, that's actually a really good question because it's becoming more popular now. And especially we had approximately 5 or 6 test days. So that's a lot of effort to always come the first time, come after a week, come after 3 months, then 6 months and 2 weeks thereafter. So that's a lot of effort. However, we actually also included so we give medication for 3 months and then we ask them to come back. And then we give more medication. And that's also to keep them a bit involved in the study because this might also increase dropout, that if you just give them a whole big batch for the 6 months, then maybe they lose interest or contact. And also after 3 months, you can look through the diary and say a little bit, okay, you've been taking it. Sometimes you've forgotten it. Is there anything that could help with that? Because we also give a medication box in which they can put the pills. However, I don't think it's an odd idea to make a clinical trial remote testing. But I did notice that the individuals in the study really enjoyed the in-person contact. You really get to know someone over 6 months. I'm not saying that cannot happen in remote testing. However, it's easier in a face-to-face setting. It would be interesting to set up the first remote clinical trial. I'm not sure right now if there's any that are doing that. Maybe in COVID that really started going, but I'm not sure. But in this, we didn't even think about it, but that's actually a really interesting topic. Back to the other question, you mentioned, well, the VLT is our main outcome. Can this test really detect all the changes that are maybe happening, or are there any subtle changes that might be happening? I'm convinced that there's also other subtle changes that are happening, and just doing a VLT, it's not enough. That's why we also ask the caregiver some questions. How are they doing in daily life? How is their mood? How is their well-being? We also ask the participant the same questions. How is your mood today? Did you have suicidal thoughts? Even those extreme questions, we still ask them because there maybe we will find a subtle change in just a random questionnaire that can also change. However, we believe in regards to memory that memory should either stabilize or improve with this medication, so we need a neuropsychological assessment as a main outcome. And I believe the 15-VLT is very sensitive, especially the delayed recall for this topic. So I do believe it's a good main outcome. However, it might be that we might miss some subtle changes, but we hope we have kind of put everything together in a way that we can catch everything. Also, when you look at other clinical trials, when you start a clinical trial, you take that as an example. So you look at others, what are they doing? What are they measuring? A CDR, MMSE, a VLT, and you look at how other clinical trials are publishing and what is their outcome. So you compare it a bit. So maybe that's also a way to start up a clinical trial you compare to other clinical trials.
Yeah, but maybe just to dig a little bit deeper again in there, because my question is like, how do you see the future and the integration of new innovative, measures, let's say digitalized in trials, because they might capture more subtle changes than classical testing, paper pencil testing will capture. Do you think there will be, more of this? Would you consider in a future trial you design maybe other instruments than the ones we're using right now?
I don't believe that the features, also the way we did it, is ready to implement yet. However, I think more studies are needed to see whether they should be implemented. I mean, we're talking about not implementation in the clinic or memory clinic, but we're talking about implementation in research. I, for one, actually also wanted to implement the app for the 15-VLT in this clinical trial. However, it was not possible due to time constraints. So that was unfortunate. So I do believe that the future will be more digital, and we will implement these in research. However, there is still it's still lacking, but I think it's coming.
Okay, thanks. I have time for a second question here. It's again about the same. No.
Yes, a short question.
Short question. I was interested in the criteria of the participant selection here for the trial protocol, because I think it will be quite broad, you know, regarding age. So I think you will end up with a very heterogeneous study population. Also, there will be MCI and mild, moderate dementia included. So how are you expecting to, you know, encounter the variability in results? Like the challenges might arise. How do you address them, or how is it planned to address them? And why did you not go for example, just MCI, a smaller, larger population?
That's an important question, because actually we always receive the opposite criticism, that it's not broad enough. You're very focusing on one group, and how can you generalize this to the rest? So it's also a factor of, you want your population to be homogeneous and not too heterogeneous, but then you might be missing other people who will take this medication as well. So it's a bit hard to find a middle ground. In the end, MCI only is a very important thought, and I think including both diagnoses even broadens the aspect more, that in a way we also look whether we can still intervene at a later stage. How we will address this is probably pull them apart as groups. So we will look at the diagnosis and statistically check whether individuals with MCI responded different to the medication than those with mild dementia. We did not include those with moderate dementia.
Okay, thank you.
Thank you very much for the opposition. And we will continue now with Dr. Arthur Korten. He is working as a neurologist at the Laurentius Hospital in Roermond.
Thank you, Paul Rector, and thank you, the supervising team, for having me here for this wonderful task as member of the assessment committee. And compliments to you, of course, Nina, standing here as a PhD candidate today, and yeah, with all the scientific work you have done. I work as a neurologist in a general hospital, so not in a university hospital, and it's very reassuring for me to see how much scientific work is done to expand our knowledge. So that's very nice. Also here in Maastricht, where already back in 86, the first memory clinic started. So that's quite some time ago. Yeah, you can imagine, as a neurologist, I also work in the field of clinical neurophysiology, and my interest was in your use of EEG and event-related potentials in your studies, mainly in chapter 5 and 6, already mentioned here. And I wanted to talk to you about the use of this technique of investigation. You write about results, and you mention all your results in tabular form, very nice. You mentioned units for latency and amplitude, that's also very nice. But eventually you conclude that there was no effect on the EEG measures and the ERP measures when you compare the people with CELTAP versus placebo. Yeah, the use of event-related potentials is well known in cognitive research. You will know that, but it also has possible pitfalls, and that's what I wanted to discuss with you. And the first thing is, I would like to comment you on, is that EEG and ERP is looking at waveforms and interpreting these waveforms. And I looked for waveforms in your presentation and also in the thesis, but I couldn't find them. And I would like to hear from you if we are missing that, or if that's not necessary. So that would be my first question.
Esteemed opponent, thank you for your compliments and thank you for your question. So you are actually interested in whether the waveforms could say something about cognition. Is that your question? Or why we did not mention any waveforms?
No, I think you mentioned something about waveforms because the potentials you report in your studies, they are related to the waveforms. But when you look at waveforms and you denominate potentials, you want to see how you did that, and that's why you need a waveform. Perhaps that's at least my opinion, working in the field of neurophysiology. So yeah, that's what I mean with the question. But maybe you can comment why you don't think you need that.
I don't know if we don't need that, to be honest. It's not my expertise. It was also someone else who performed the analyses for the EEG. However, the ERPs are also usually used within the same literature. It's what people look at, such as the P600, which is very famous for cognition, or the P3. And that's also a good way of communication. So I can't really answer on whether waveforms would have been, or it could have been interesting also to mention in this.
Okay, I understand. Maybe in the future that will change. There is a lot of discussion about only using the peaks or maybe using the waveform as a whole, but we'll leave that. Yeah, you also talk about artifacts in the registration of the ERPs, and you tell us that you correct for artifacts in the 1 second after the stimulus. That's what you mentioned in the 2 chapters. But when you look at the literature concerning ERPs, it's also very important to look what is happening before the stimulus, to have a good signal-noise ratio, to use these ERP potentials in a correct way. Yeah, you explicitly mentioned that you looked in the second after the stimulus. Was that a choice you made with certain reasons, or maybe you can clarify that?
That was, I think, not a specific choice we made with certain reasons. No.
Okay. Last question. Also about this difficult subject, there is also the problem of anticipatory activity in the ERPs, and yeah, the literature says that activity can contaminate your results. Yeah, is it possible that maybe the fact that you didn't find a relevant result, comparing placebo to CILTAP using the ERP, that has something to do with not compensating for that?
That could be definitely a possible reason. The question I then have, what does it say if you have brain changes related to treatment, but that they do not reflect necessarily on the cognitive tasks? So you would have to have a look at that interaction effect, but that could definitely be a reason why we maybe did not see systemic brain changes due to EEG.
Okay, thank you for your answers. My word goes back to the prorector. Thank you.
Thank you. The opposition will be continued by Dr. Dimfie Indebraak. She's working at the Department of Clinical Neuropsychology, at Maastricht University.
Thank you. Dear candidate, first I would like to congratulate you on this thesis, which addresses important topics related to the diagnostic process of cognitive disorders and dementia, very important subject. And I would also like to congratulate your promoter and co-promoter as well. And of course, I would like to go into more detail into chapter 2, which you describe the diagnostic and prognostic value of the digit span test across the diagnostic spectrum. And you found that 2 subtests significantly differed between the stages of cognitive decline, and that the backward and sequencing subtests showed significant predictive value for dementia progression, and therefore is of value in the early prognostic or early diagnostic process. And as a clinical neuropsychologist, I see a lot of patients that come into the clinic more early and want to know if their subjective complaints are an indication of early dementia. And in my experience, working with a lot of patients, even in a later stage of dementia, the working memory scores are still intact, at least as you compare them with the norm scores. And that memory test, like the 15-word verbal learning test, are more, we talked about that earlier, are more sensitive and informative in the diagnostic process. Of course, not only that test, but it's more, at least more informative. So what I see in the clinic is that the digit span is not always deviant, or mostly not deviant, whereas 15-word verbal learning test is. And could you help me explain how the digit span subtest scores could help me in the individual diagnostic process, or me or any clinical neuropsychologist in this case? And should we be alerted by below average scores on a digit span, for instance? Because I know that a lot of conditions do influence working memory. And so, in other words, how can we apply our findings in clinical practice?
Esteemed opponent, thank you for your compliments and your question. That is the eventual goal, to see that we do research and that actually the clinic can do something with those findings. So the memory clinic is a very heterogeneous population. So individuals that can come in, even if it's very early, it's hard to know what the prediction indeed is, and also what the reason is for cognitive issues. So, and as you mentioned, they come in from different backgrounds, and working memory is usually not something we see impaired early in at least Alzheimer's disease or the classical cognitive impairment. However, we do know that, for example, depressive symptoms, so depression or major depression or anxiety symptoms can influence cognition, and that the digit span is something that could be sensitive to this also, because it measures working memory. Also in a population of brain-acquired injury, this is also a test that maybe is more valuable than someone in Alzheimer's disease. However, we don't know the etiology when someone comes in with SCD. So my suggestion, not as advice, or just I think that my opinion is that when someone comes in the clinic, and as you mentioned, the 15-VLT is very popular because it's more sensitive. However, I think we should measure each cognitive domain that we have. So that includes language, attention, memory, all of those. We should actually just make a repertoire of that. And for each cognitive domain, we should have a test that measures the cognitive domain in which the digit span is the most famous for the working memory.
Yeah, and would you argue that it's a memory test, or is it more attention or executive test? Because I think that would be a nice discussion as well.
I think it's more attention and executive, that you actually can separate the subtests from each other. As we saw, the forward test, actually, if you would, and you mentioned me, should you be worried if someone fails this test? I think one should maybe be cautious if someone fails the forward test, because it is actually just a preparatory test for the next subtest. So if someone would fail the forward test, then maybe there might be something fishy.
Yeah, but I think in the clinical situation, we almost never encounter that someone doesn't pass that test. And even if in a geriatric population, it's often still intact. So that's a difficulty, I think. Yeah. But
Dear Ms. Possemis, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. The mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. To the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Nina Possemis. The degree committee here present has assessed the quality of your thesis and your defense. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Verheij is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
So traditionally we will start with the oath, the scientific oath. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
I promise.
By the authority vested in us by law and in conformity with the decisions of the committee here present, I hereby confer upon you, Nina Maria Possemis, the degree of doctor and grant you all rights attached to by custom and law. And as evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university. We have a second certificate which will be handed by the co-promoter Ine Ramakers.
Well, Nina, another certificate. On behalf of the Research Institute of Mens, I am entitled to hand out this official certificate. The owner of this certificate has received training in a broad range of academic skills and has gained experience that is relevant for a successful career as an independent scholar. Dear Dr. Possemis, dear Nina, today I have the honor of being the first to congratulate you on your well-deserved title. You have produced a remarkable book of scientific studies focused on the diagnosis and treatment of memory disorders. And since you joined our Research Institute of Mens, your enthusiasm for science quickly caught the attention of colleagues of Division 3. As a talented graduate student, you received a grant from Division 3, marking the beginning of your PhD journey. ZonMW approved a generous grant for Division 3, which allowed us to appoint you for the subsequent years. The project investigated the effects of the PDE4 inhibitors on cognition, linking the expertise of Division 3 with the knowledge of cognitive disorders and dementia in a memory clinic population from Division 1. And this required you to be versatile, with a strong understanding of the biological mechanisms of these drugs, with laboratory skills, and the ability to empathically engage with people with memory issues and dementia. And you have proven today that we were right in expecting you to excel in this role. The project encountered several challenges. The start of the project almost coincided with the start of the COVID pandemic. The METC had other priorities. The outpatient clinic was closed and later reopened at reduced capacity. The supplier of the study drug faced its own COVID-related problems, and our patients were understandably hesitant to visit the hospital. And even after the pandemic, enrollment was slower than anticipated on various reasons. Yet you persevered tirelessly promoting Romima. You surprised us with beautifully designed Romima cups. You created Romima flyers, and Romima had its own website. In 2021, a significant article appeared in the local newspaper, the Limburger, and it was a nice initiative to support recruitment. The timing, however, was unfortunate, and it was published just before your holiday. And consequently, many interested individuals applied while you were away. Yet you managed to respond personally to every application from a village somewhere in the mountains. Enrollment progressed slowly but steadily, thanks to, in part to the Zuiderland hospital support. And another article, this time in a Belgian newspaper, generated over 100 responses. And we would have loved to receive such an interest at any point during these 5 years. But again, the timing was less than ideal because it was just a few weeks before your maternity leave. But Nina wouldn't be Nina if all those 100 didn't get a personal reply before she left. In 2023, you welcomed your adorable son, Aris. We joked that you now had a second child. After all, your first was Miss Romima. During your maternity leave, you had to step back from Romima at least a bit, which was not easy for you. You prepared your leave perfectly, providing Florence, Daphne and me with training sessions per test day. You wrote very detailed protocols for us, essentially a Romima for dummies. And even while on leave, you remained accessible and kept a watchful eye on the progress of the project. You were passionate in doing clinical research, and the participants touched your heart as you became genuinely involved in their stories and in their concerns. During testing, you built a meaningful relationship with them, and during your leave, participants often inquired about you. How is Nina? How is the baby? And what's his name? There have been unexpected circumstances along the way that also have brought changes and that you had to deal with. But you found your way through, always keeping your scientific goals in focus, and your colleagues regard you as a reliable team member, always available for support. Even during holidays, you provided your mobile number in your out of office. And in addition to Romima, you engaged in several other projects, such as the Deep Spa project, focused on advanced speech analysis, the biobank cohort of our Alzheimer's Center, of course, and you analyzed CILTAB data. This demonstrates your flexibility and your willingness to explore diverse aspects of your field, culminating in this impressive dissertation, comprising of 6 scientific papers, most of which have been published or accepted in esteemed journals. Beyond your primary project responsibilities, you was always available for other tasks. Whether it was quickly photoshopping last-minute Christmas card for our ACL client panel, classifying medication of the ACL cohort, conducting deep spa phone assessment during the pandemic, or assisting with experiments in Division 3. You also played a vital role in representing PhD students. Although assigned to Division 3, you quickly became the PhD representative of Division 1. and you served as a PhD representative of the NANS network, and you represented all men's PhD students within the UM committee. You organized pizza meetings, and you contributed to the ACL newsletter, becoming a well-known figure soon. And you worked together with many people in Division 1 and Division 3, as well as with FPN, psychopharmacology, with our clinical colleagues from the memory clinic, with Key Elements in Saarbrücken, showcasing your exceptional talent for collaborating and networking. Additionally, you participated in teaching activities, both clinically oriented, but also practical courses at Division 3. And you supervised several interns at our BBACL cohort. You took part of the affective neuroscience course, initially held in villas in Florence, but you ended up in a monastery in Leuven, where your team won the award for the best assignment of the summer course. You presented your research findings at important conferences such as Alzheimer Europe in Helsinki or 3 weeks ago in Geneva. You approached your work with great care and dedication. The results of the monitoring visits by the CTCM consistently reflected that everything was in perfect order. However, the monitor did have a challenging time tracking your key file, which seemed to require a code for the code. So truly super double-proof GCP. In summary, you are an exceptional hard worker. Quiet, yet powerful, and appreciated by all. You possess deep knowledge, independently managed the entire trial, and made informed scientific and ethical judgments. You were attentive to your colleagues and to our interns, managing to juggle numerous responsibilities alongside your own project. And during this time, you also got married and welcomed your son Aris. It has been a pleasure working with you, and I consider it as an honor to have been your co-supervisor. I'm delighted that you will continue our collaboration in the Alzheimer Centrum as a postdoc researcher for the time being. Once again, I would like to extend my congratulations also on behalf of Hans and Arjan on this remarkable and well-deserved achievement. And I also wish to acknowledge your family, your in-laws, your husband Halim, and little Aris. Enjoy your title, and congratulations.
Dear Dr. Possemis, also on behalf of Maastricht University, I want to congratulate you with the degree, but also really the admirable way that you conducted an always difficult phase 2 trial during challenging times. And then I'm even more surprised to hear what you did in addition to all this scientific work. So really admirable. We're nearly there for all of you to congratulate Nina, but what we want to do is first the promotion team, the opposition, Nina, and perhaps her family, to take a picture here, because then also the opponent online can be present. Thereafter, we thought it might be nice to have an additional picture on the steps in the aula. But please, audience, do not wait. Go already to the rafter, take a drink, and thereafter Nina will come and you can congratulate her. But for now, I will end this academic ceremony.
Hey, Yogi.